Quantitative multiplex co-immunoprecipitation, or QMI, enables users to simultaneously measure hundreds of binary interactions in a predefined protein interaction network. By performing multiple co-IPs simultaneously in the same lysate, bioinformatics techniques can then examine how groups of co-associations change in a coordinated manner.
The assay is time-consuming to develop, but once a QMI panel is in hand, network-scale data can be collected about signal transduction systems in a relatively simple overnight assay. QMI requires the accurate pipetting of different samples and reagents into 96-well plates. Careful notes should be taken when setting up and running each assay to prevent errors.
For sample preparation and immunoprecipitation, begin by lysing the sample in a suitable detergent with protease and phosphatase inhibitors for a 15-minute incubation on ice. At the end of the incubation, spin down the sample to remove the membranes and debris, and perform a BCA assay on the supernatant to determine the protein concentration according to standard protocols. After normalizing the protein concentrations, prepare a magnetic bead master mix that contains approximately 250 magnetic beads of each class per well. Use magnetic separation to wash the magnetic bead mix 2 times in Fly-P buffer before resuspending the beads in 20 microliters of Fly-P buffer per sample. After thorough pipetting, aliquot 20 microliters of beads into 1 ice-cold microcentrifuge tube per sample and add equal